All right, welcome everybody. Thank you so much for joining us. My name is Andrea Decker and I have the pleasure of working for the Fleet Science Center. And we're excited to welcome you for another talk of our special talk series, Live Longer, Live Stronger, in partnership with the Sanford Burnham Prebys Medical Discovery Institute. And today, as you know, we're going to talk about how the heart ages. And I'm very excited to have Dr. Karen Occor join us today. With her presentation. Just a couple of things before we get started. As you might have seen in the PowerPoint presentation running before we got started, this is a Zoom webinar, so the chat function and your camera and audio will be disabled, but you can engage with us through the Q&A function. At the bottom of the screen, you see two little speech bubbles, and if you click on that, it opens a window where you can send us questions and comments, and I will keep an eye on those. And there will be time for Q&A after Dr. Occor's presentation, but you don't have to wait to type your questions until then. You can type it in and send it off at any time. This way you don't forget what you wanted to ask. So at any time, please feel free to send your questions. And at the end, when we get to the Q&A, I will open a little poll, a little survey. So if you could take a minute to fill that out while we answer your questions, that will be wonderful. We always love to hear who our audience is and what we could do better. And these surveys do help us a lot with that. So I appreciate that. There will be two more talks in this series, as you might have seen in the PowerPoint presentation. We have one on vision loss and then on bone strength, the next one April and then May. It's always the first Wednesday of the month from 2 to 3 p.m. And then I have a special announcement, and you will get an email about that. Everybody who joined us for any of these talks will get an invitation to join us on May 26th, which is the last Wednesday in May, from 2 to 3 p.m. to join us when we have all the speakers back in a regular Zoom room, and we will put each speaker into a breakout room, and then you can visit each speaker on the topic that you're most interested in, or switch rooms and talk to as many as you want to. So that special invitation is going out sometime soon, so keep an eye out for that. And I think that concludes my little introduction. And I don't want to take more time away from the talk that Dr. Occor has scheduled for us. So Karen, why don't you take it away?
Thank you very much. I appreciate the opportunity to present the work of our lab, and I'm going to also show a little bit of the work of our collaborators that we're working with to try to understand the genetic molecular basis for atrial fibrillation. And let me share my screen. Okay. So we use, what I'm going to tell you about today is how we use the fruit fly model system to understand the genetic molecular basis for heart disease in general. And we've more recently been turning our attention to atrial fibrillation in particular. So despite years of research, heart disease is still the #1 cause of death in the industrialized world. 1 in 4 people die of heart disease. And it's still a very big black box as to exactly how all of the risk factors that we now know about, how they work together to cause heart disease. And we use the fruit fly model system because it's a fairly simple system, but has some very important characteristics that are similar to humans. So basically, what we want to know is what causes heart disease and how can we leverage the fruit fly to learn about it. Well, an important critical component is fruit flies have to have a heart, or your organism has to have a heart, and fruit flies do, and you'll get to see that. They're small, so we don't need a huge facility to keep them. We can keep thousands of individuals on the end of a lab bench. They importantly have 75% of the genes that cause disease in humans. And that means if we want to understand how those genes cause disease, they're there available for us to manipulate in the fly. And we have, as you'll also see, lots of genetic tools that allow us to manipulate flies. And probably the most important feature of fruit fly is summarized in this short little video.
fly, huh?
So, what's it like having such a short life cycle?
It's great, mister. When I grow up,
I'm gonna wish I had done more with my life, Sonny.
So let me get back to my presentation. That summarizes in a nutshell one of the big advantages of the fruit fly system. It has a very short lifespan. So if we want to understand how aging impacts heart disease, we don't need to wait decades if we were looking at humans, or even years if we were working on mice. We can know in a few months, because a fruit fly's life is 1 week of a fruit fly's life is about equivalent to 10 years of a human lifespan. And so flies live around 7 to 8 weeks, so 70 to 80 years in human terms. And so it turns out to be a very nice system if you want to start looking at things that relate to age. But how do we know, even so far, what the risks are for heart disease? Where did that information come from? How do we know age affects heart disease? Well, it came from a very interesting early study called the Framingham Heart Study. And it was a study that drew 5,200 men and women from Framingham, Massachusetts, and followed them over the years up to current day, in fact. And also there's now other studies done on their offspring and their grandchildren. And the idea was to try and track who developed heart disease and what the things that they ate and did were that might have contributed to different levels of heart disease. So it turned out to be a very important study. It gave us lots of insight. And what they were tracking were things like high blood pressure, increased cholesterol, smoking, diabetes, family history, all the things that we now know or claim we know are involved in heart disease. But they were tracking them to try to show there was a correlation. So I'm just going to share with you one of their graphs. And I promise this is the only graph you're going to see. There will be no test afterwards. But it illustrates some really interesting pieces of information. So what this is showing you is the incidence of heart disease over time. And it's showing how if you increase the number of those potential risk factors, you get subsequent increases in heart disease incidence as you add on risk factors. So that was one thing that they learned from this study. Another thing that was quite unexpected, I think, was that if you match risk factors, for equal numbers of risk factors, women have less heart disease than men. So that was a fairly novel and unexpected result from this. But also, if you have no risk factors or minimal risk factors, you still have an increase in heart disease with age. So that was an important additional finding from this early study. But we don't need to look at that study to really see that, that age is related to heart disease. Now, if we simply look at the incidence of hospitalization for arrhythmia, you can see there's a fairly high incidence in the eastern half of the country. If we match that with the percent of population over 65 years of age, you can see that there's a considerable amount of overlap, which again is evidence that something in the aging process is associated with heart disease. In addition, we've also been able to correlate from the Framingham study, as well as just epidemiological information, that obesity has plays a role in the incidence of heart disease. If you again compare the hospitalization rates, they overlap fairly nicely with the incidence of obesity in the country. So of arrhythmias for which people are hospitalized, atrial fibrillation is the most common form in the country. And the incidence is increasing by 3% per year. And this is in large part due to the fact that the population is aging, but it also has to do with increases in obesity and other risk factors. Importantly, an important thing to understand is that for atrial fibrillation, while we have some palliative treatments, there is no cure. There is no treatment that makes it go away, at least not permanently. So it's an important disease burden for the world, in fact. So what is atrial fibrillation? Well, here what you're looking at is a schematic of a heart, and it's showing the normal progression of electrical activity from the atria, which causes them to contract, and then to the ventricles, which subsequently contract and pump blood to the whole body. So this is what's called sinus rhythm. It's a normal contraction proceeding from the top of the atrium to the ventricles. In atrial fibrillation, for reasons that we still don't understand, this electrical activity is disrupted. It becomes chaotic, and that chaotic electrical activity doesn't allow the atria to contract correctly, and it can also impact ventricular contraction as well. But the primary effect is in the atria. Because the blood is not pumped through the body effectively, the symptoms of atrial fibrillation include, of course, fatigue, because you're not adequately supplying oxygen to your body. But interestingly, one of the main problems that is associated with AF is stroke. And what happens to cause the stroke is that this ineffectively pumped blood in the atria tends to pool, and it can pool behind something called an atrial appendage, which is kind of an appendix for the atria. It's not really a useful piece of tissue, but it prevents the blood from moving around effectively. And then when it pools behind this appendage, it can tend to clot. And that clot then ultimately can be expelled and go to the brain and block blood flow there, causing a stroke. So that's one of the main problems with atrial fibrillation. And so when you're diagnosed with AF, you're usually put on blood thinners, which helps to prevent that stroke. Untreated atrial fibrillation can lead to heart failure because this chaotic electrical activity in the ventricles begins to cause electrical remodeling in the ventricle, in ventricular tissue, and it can lead to other kinds of heart disease as well. So it's a serious problem. And as I said, it's increasing in incidence and with age in our aging population. So this was all information that I've told you about that was generated from correlational kinds of studies. But with the sequencing of the human genome, which was completed in 2003, we now had an opportunity to start to look for a genetic connection for heart disease. And again, coming out of the genetic analysis of these patients in the Framingham study, in 2009, the first genetic variant associated with AFib was identified. Since then, a whole bunch of studies have been done to identify many more genes that have at least been linked from patients with AFib to the disease. We have used the genetic bioinformatic tools that are out there to see what the connections are between these 200 potential AFib causing genes. And this is the hairball that we come up with. Now that we know what all these genes do and they've been studied, we can find out that some of them are connected, but to try and untangle this mess of genetic interactions to really understand what's going on in AFib is going to be a Herculean task. And then you can't forget that smoking, obesity, age, all of those other sort of nondescript monikers impact what those genes are doing. So AFib is a multifactorial disease. There's lots of things that impact the development and progression of AFib. So how are we going to figure this out and come up with the drivers or important drivers of the disease? Well, that's where the fly comes in, of course. So I promised you I was going to show you the fruit fly heart, and here it is. Should be beating. It's not beating. Why is it not beating? There we go. So the fruit fly has an abdominal heart. It's a linear heart tube. It has 4 chambers that are separated by valves. But it, unlike your heart, which also starts out as a linear tube, it doesn't twist and form the 4-chamber heart that we have. And it stays linear even into adulthood. We can take these fruit flies and we can dissect them and get a higher image, a higher magnification image of that heart tube. And if we do this with a very high frame rate, high speed movie, we can get a lot of information about the function of the heart just from this movie. For example, we can take an electronic slice through the movie, and then we can line up those slices like slats on a picket fence, and we get something called a motion mode or M mode. And it's exactly like what doctors do with echocardiograms to get the same information about how well the heart contracts and the rhythm in which it contracts. So here you can see the heart edges contracting and relaxing. Whoops, did not want to do that. You can see them contracting and relaxing in a very rhythmic kind of way. So we've got now a model system that we can use to be able to understand heart function, at least in the fly. But is that going to have any relation, any relevance to humans? Well, I think it does, because remember I told you that cardiac arrhythmia increases with age? Well, this is also true in the fly. Here's a young 1-week-old fly with a very nice rhythmic heart pattern. And here's a 5-week-old, 50-ish-year-old fly, and you can clearly see that its heart is arrhythmic. So like humans, arrhythmia increases with age in flies. In addition, obese flies develop heart disease. So here's a normal food-fed fly heart beating very nicely. And here's one that was fed 30% coconut oil in its diet. Now, 30% is the recommended upper limit by the USDA for fat intake by humans. But after only 5 days on this 30% coconut oil diet, you can see this heart is quite irregular. It's not contracting properly. It's really an unhappy heart. Now, just to put in perspective what a 30% fat content in your diet is, if you weigh 140 pounds, that's between 56 and 70 grams of fat. And this is what 70 grams of fat looks like. So on a daily basis, it's not that hard to get this amount of fat in from your diet. So we see that there's arrhythmia with obesity in flies. But interestingly, we also were able to show that parents' diet matters. If you feed a parent a high-fat diet, so they have cardiac arrhythmia, and then you let them have offspring, and you look at heart function in their children, even though the children are fed a normal food diet, they still have heart dysfunction. In fact, it takes 2 generations for their heart function to return to normal. Now you might say that, yeah, what does this have to do with humans? Well, we can't really force people to eat particular diets, but there are some sort of serendipitous experiments that have been done on people. For example, during the Second World War, there was a blockade by the Nazis of the Netherlands, and they ended up running out of food. Essentially, they had to subsist on about a 30% of their normal diet or normal caloric intake during this 5 or 6 month period under which they were blockaded. And scientists following this period went and examined the health and lifespan of the offspring who were conceived and gestated during this period. So in utero, forced to be exposed to this very low calorie diet. And what they found was that the offspring were smaller than average, And in addition, as they got older, they were more prone to diabetes, metabolic syndrome, as well as heart disease. So even in humans, there's a number of these kinds of examples which show that the diet of the parent, it does matter on heart, with respect to heart function in offspring. So again, we have a parallel between flies and humans. In addition, so I said that there were these genes that had been associated with atrial fibrillation already, just correlated from genomic information of AFib patients. Well, one of those genes, if you mutate it in the fly, you amazingly get a heart that fibrillates. So in addition to diet, age, gene mutations that have that are related to atrial fibrillation in humans also cause a fibrillatory kind of heart function in the fly. So given all of these sort of parallels, we feel pretty comfortable that we have a model system where we can now exploit this genetically manipulatable model and aging model to try to understand how these things all work together to cause atrial fibrillation. Okay, so how are we doing this? So we are combining forces, my lab as well as another fruit fly lab run by Rolf Bodmer. We're combining the fly models that our labs have identified and generated with a model identified and developed by Alexander Kolas, also at SVP. And he uses human-induced human cardiomyocytes. So basically you take skin cells and you make them become essentially a heart muscle cell. And here you can see an example of how this looks. These cells are rhythmically contracting. And our idea is to try and use both model systems to get at common genes that are likely to have been conserved in humans that cause these cells to beat arrhythmically or cause this fly heart to fibrillate. And we can do it in a relatively high throughput manner. So Alex can take these induced cardiomyocytes and put them in plates that have 396 wells, and he can manipulate those cells, and I'll show you how in a second, and then look to see what effect that has on cardiac contraction in his cultures. We can take a high throughput variation of our fly model system that was developed by Geo Vogler in Rolf Bodmer's lab, using a fluorescent reporter that can be seen through the cuticle of the fly. So you don't have to open it up. And you can then more rapidly start to look for fibrillatory kinds of responses in the fly when we manipulate genes. And so our intention is to test those 200 AFib related genes, and the list is growing, in both systems, and identify ones that are having similar effects in both systems to come up with underlying gene networks we can pull out of that hairball I showed you, and then go back and do further tests either at the patient level or even in other model systems. So how do we test individual genes? Well, both systems use a similar method. So as you may know, instructions for how to make a cell are contained in the DNA in the nucleus of any cell. And those instructions get translated out as sent out into the cytoplasm of the cell as a messenger molecule with the information contained in that DNA. And that information from the messenger is then used by the cell to make, for example, heart muscle proteins. And those proteins then are incorporated into the components of the muscle that allow it to contract. And so if there's problems in making this heart muscle protein or any other protein, you should be able to see it in terms of change in the function. So what we can do in both systems is we can introduce essentially an interfering RNA that knocks out this message. And that means that the heart muscle protein or any other protein we want to knock out does not get made. And so we can go through, and in both the fly, we can perform this kind of what we call gene knockdown, and we can do it just in the heart, and we can then look to see what the effect on heart function is. Similarly, in Alex's system, he can put it into those wells containing these induced cardiomyocytes, and then this will allow him to look at function and contraction in human cells. And in both systems, we can do this in a relatively high throughput manner. We've done this previously for other projects, and we've been able to screen on the order of about 1,000 genes in a year, which means we can do quite a large amount of trials for these potential AFib genes. So as proof of principle, we've started on our analysis of these 200 potential AFib-related genes. And what you're looking at here is that hairball that I made of trying to network all of these genes together. And we've tested all the ones that have circles around them. So the red circles means we've tested them. The yellow circles means not only have we tested them, but they did similar things in both our fly system and in the human cardiomyocytes. And so we've taken the ones that we identified as hits, and now we've used new bioinformatic tools to try and network them together. And this is what we come up with. So this is a much simpler hairball than the one I showed you. And actually, there's a reasonably good chance that we will be able to get through all of these genes and test them to see which ones actually do interact and pull out the relevant drivers, if you will, for atrial fibrillation. But we're already focusing on one subnetwork, which is shown in this dashed box here. And that subnetwork, if we pull it out, is shown here. And you can see it contains two of our hits that are AFib-related genes that have similar effects in fly and human cells. It also contains a couple other AFib-related genes we haven't tested yet, but it also includes a bunch of other ones that we might never have thought to pull out and include in our analysis. But it is this particular network that we're currently focusing on to try and see if we really do have connections between these genes that cause fibrillation in either one of our systems, and also that maybe connect the effects of obesity and diet to the development of atrial fibrillation. And the reason for that is because one of these genes, which is a desaturase enzyme, that gene is already a drug target against obesity. And it is involved in making monosaturated fats from saturated fats. So good fats from, good fats from bad fats. And so it's a uniquely interesting gene that may allow us to connect genes that we think cause AFib that are not enzymes, that are things called ion channels, with muscle proteins. This is a muscle protein, this is an ion channel, and this enzyme might provide the missing link in terms of creating a substrate for atrial fibrillation with age. And so our system then will allow us to look at both the effects of knocking down these genes with age, and also we can go and look and see what happens if we manipulate these genes. Can we prevent this kind of diet-induced cardiac arrhythmia? So we have a very nice system, I think, that will allow us to look at the role diet and age in the fly, validate in human cells, and hopefully come up with some information that allows us to begin to target a treatment for atrial fibrillation. Because as of now, there is really no known cure, as I indicated. The only treatment right now involves a surgical procedure that electrocoagulates those cells that are causing the chaotic electrical activity. And typically that will work for a couple of years, but then the tissues around that coagulated portion of the atria begin to become electrically chaotic again. So there's really no permanent cure for this particular disease. So we hope eventually to be able to go through all of these genes, as well as the growing list in our system, and tease out the important underlying pathways. And with that, I would like to acknowledge my funders. So we have funding from the American Heart Association, National Institutes of Health, from our institute. And interestingly, we've been able to use this model system for experiments aboard the International Space Station. So we actually have funding from NASA to try and understand the role of microgravity and how it impacts heart function. And the labs that I've been talking about, my lab with the two people from my lab who are importantly involved in this project, Rolf's lab, I talked about work by 3 of the people in his lab, and then Alex Colas' lab as well. And with that, I thank you for your attention, and I'm happy to take any questions.
Wonderful. Thank you so much, Karen. I really appreciate it. What a fascinating topic. Slightly scary, I'm not going to lie, as I just celebrated my birthday in February.
None of us are getting younger.
That is certainly true. So we have one question already, and I'm going to read that. And it is there an agreed upon definition of heart disease? For example, does it depend on the presence of coronary artery disease or something like that?
There's all different kinds of heart disease. Our focus right now is on arrhythmia, which has to do with how the electrical activity that causes your heart to contract can be aberrant. But then there's structural heart disease, where your heart can be dilated, it can be hypertrophic. And then coronary heart disease is yet again another category where the blood supply to the heart is impacted for different reasons. But obviously, if you don't get oxygen to your heart, it's not going to be able to contract properly. So there's all different kinds of heart disease. And as I said, we're focusing right now on atrial fibrillation, but we also have a couple other projects involving congenital heart disease and also cardiac hypertrophy.
Thank you. Next question is, if flies and humans share 73% of genome, what are chances that the smoking gene is in the 27% we don't share?
That's a good question. And yes, it's true. There may be 1 or more genes that we miss because there isn't 100% overlap. On the other hand, we can always take this out of the fly if we have a pathway we think may be important, but we don't have all the pieces in the fly. We can take our analysis out of the fly and put it into the human cardiomyocytes. We also use a vertebrate zebrafish model that allows us to take it into something that has a few more shared genes with humans. And ultimately, we can work with our collaborators. We have several who work on atrial fibrillation in humans, and we can go and start probing the genome data from those patients to see if that pathway is altered in terms of different genetic variants that are found in AFib patients. So we have a lot of tools we can bring together to get at that possibility.
And just to clarify, cardiomyocytes are heart cells in a dish, right? That function a little bit like a heart. I've heard something like that for the brain. They have neuromyocytes. And it's kind of like a little mini brain in a dish. Is that the same for the cardiomyocytes?
That's correct. So cardiomyocyte is the functional part of the heart. So it's what contracts when your heart pumps. There's lots of other cells in your heart, but what we're interested in, because it's the workhorse, it's what makes the blood go round. We're interested in the cardiomyocytes. And so what Alex has developed is a system to be able to induce skin cells, human fibroblasts, to become cardiomyocytes. And he can even make them into specifically atrial or ventricular cardiomyocytes, because there are slight differences in the properties between those two parts of the heart. And so they don't have the full capability and protein complement of a true heart cell, but they are a pretty close equivalent, and they allow us to test the involvement of different genes in producing a rhythmic contraction.
I think that's amazing, because then you don't have to run any risky experiments in patients, because you can test it out on those
Right.
first.
There's also another thing that we can potentially do is when you're treated for almost any kind of heart disease, that atrial appendage that I mentioned earlier that causes blood to pool is usually taken out for that very reason, because it's not doing you any good by sitting there in your atria. And so it's usually taken out. So we can go and look at what genes are expressed in those pieces, especially from, interesting would be from patients with AFib. So that's another way we can get at the genetic and molecular pathways that might be contributing to this disease.
Amazing. I've got a bunch of other questions here. Once you identify the genes involved in AFib, what do you do with that information?
Yeah, so once we have identified the genes, then we're going to try to figure out how they interact in order to identify how to, as I said, how do we connect aging and diet and genetic variants together to cause a disease. And so what we'll do is we'll start looking at their function in young and old flies. We'll feed them a high-fat diet and see if we can manipulate the gene and prevent that cardiac dysfunction in response to a high-fat diet. We can also knock down several genes at the same time. So if we think two genes are interacting together, we can directly test that. We might not see any huge effect from one gene knockdown, but when we put them together, we might see all of a sudden this massive atrial fibrillation, electrical remodeling result. So those are the kinds of approaches that we're taking. And we also look for structural effects because electrical activity doesn't only affect the contraction, it can ultimately cause problems in terms of heart wall thickness and heart remodeling.
Thank you. And another question is, can you have AFib and not know it?
I think probably in very early stages you might, but I think most of the time, once it becomes a significant issue, you're going to start feeling a lot, at the very minimum, a lot of fatigue, but it's gonna, I honestly don't know how it feels to have a fibrillation attack, but it's a bit like a heart flutter, although it's a different, AFib is a different disease than heart flutter. But it's something that you would probably go to your doctor because you're fatigued all the time and something doesn't feel right in the way your heart is working, and then they would do EKGs and diagnose you from there.
that kind of leads into the next question, which is how do they diagnose AFib? Is it through the EKG? Is more needed than that?
Yeah, typically they look to see if there's a P wave, and and most of the time, if you have a problem in your atria, you lose one of the little blips in the EKG signal that you saw at the beginning of my stylized EKG trace at the beginning of the talk. So the little blip at the very beginning is when your atria contracts, and you lose that if you have AFib. So that would be, I think, the major way.
So it's at least relatively easy to diagnose, which is good news. And then can you name and describe the process used to change human skin cells into the heart muscle tissue?
Yeah, I very simply what you do is you essentially de-differentiate those cells. You make them into a more multipotent kind of cell, and it involves putting some factors onto them and incubating them. And there's other tricks that Alex has worked out. And then you try to coax them into a developmental pathway that produces a muscle cell. And in his cases, atrial and ventricular muscle cells. It's something that we do quite a lot now. They're called induced pluripotent stem cells, where we're taking them and making them into all kinds of different tissues so that we can study different tissues in a dish. So it's a relatively routine thing that can be done in the lab now. But to be able to reliably get just atrial cells and just ventricular cells, that's a little trickier, and Alex Colas' lab has perfected that.
Awesome. Amazing what you can do these days.
I know, it's so cool. You look at these things in under a microscope and they're just all happily beating together.
Yeah, exactly. I find it fascinating. Do you have a hope for timeline for when your efforts might become available for clinicians treating patients with AFib?
Yeah, so I think we're looking at trying to, or we certainly can go through all of those AFib genes in a year and a half. One of the problems, of course, is getting funding for the manpower to do that, and we're busily trying to do that now, which is why we're working right now just on sort of pilot studies in order to be able to convince government agencies to give us some money so we can go further. But I think we would be able to test all of those genes within a year, then probably another year to verify the ones that we think are interacting and work out exactly how they're interacting. And then we have clinician partners that we would go to and then along the way and check out their genomic data from their patients for signatures of the genes that we think we're identifying are involved in AFib.
So a year and a half for sure to go through the genes. And then of course it will take some time.
After.
Yeah. Well, but
Yeah.
that's still
Well, the thing is right now, there's really no other way to do the kind of genetic interaction studies that we're doing. This is kind of a first of its kind system that we're trying to push forward. It would take much longer if you tried to do this in mice, for example, if it was possible at all to do in mice in this kind of way. You'd have to be very lucky because you can only manipulate a few genes at a time in a mouse model, whereas in our models, we can manipulate almost at will as many genes as we want. Certainly in the cardiomyocytes, we can do all kinds of combinatorial gene knockdown experiments. It's a little harder in the fly, but we can still do multiple genetic knockdowns in that model as well.
Thank you for that. And part of your research focus is on the diet. And the question is, why do you focus on diet when that is under patient control? That's not necessarily something that you or a clinician could influence completely.
Well, we're focused on diet from the perspective of there's a connection between obesity and AFib. We don't know what it is, but in the fly, we can manipulate the diet, which you can't in patients. So that's a very good point. And it would be really tough just studying patients to identify exactly how diet is impacting heart disease, whereas we can rigorously control it in the fly. So for example, the high-fat diet in the fly caused that very arrhythmic heart contractions. If we feed it a high-sugar diet, we have a different, we still see some arrhythmia, but now we see more fibrosis when we feed it a high-sugar diet. So diet has some differences depending on what it is you're eating in terms of how it impacts, at least in the fly, how it impacts heart function. And so those differences may be important. And as I said, this is something we can tease out in the fly that would be impossible to do if you're studying human patients. Possible to do in mouse, but not in humans. The idea, though, is if we can figure out what the connection is, that gives us a target for treatment, perhaps not only of obesity, for example, in the case of that last enzyme that I talked about, not only in the case of obesity, but it may also have an effect on cardiac disease as well. So knowing exactly how they're connected will make it possible for us to design those kinds of effective treatments.
Thank you. And then there's a question, if somebody is experiencing AFib after a stem cell transplant of T-cell lymphoma, is that a permanent AFib, or is it just something that happened because of that procedure?
Yeah, that's a good question. So yes, often after heart procedures, they will have some transient fibrillation. My understanding, I don't know for sure, but my understanding is that that typically goes away. I don't know if that means you're at higher risk for later developing more persistent kinds of atrial fibrillation. But yeah, it is a secondary effect of heart surgery often, yeah.
And we have two questions that kind of go together. Someone would like to know what the difference is between AFib and an atrial flutter, and then if an atrial flutter can actually develop into AFib.
They are similar, but they're different diseases. So a flutter is basically the heart beating very fast, but it's still beating somewhat normally in terms of the progression of the electrical activity from the top of the atria down to the atrioventricular node. So you still get the heart contracting more or less well enough to move blood around, although there's still a risk of stroke even with the atrial flutter. Atrial fibrillation, though, is a completely chaotic progression of that electrical activity. And so it's thought to be a different disease, a different cause, just because flutter does not progress into AFib. So we think they're different diseases, and in neither case do we really understand the root cause. And that's why we're doing the work we do.
I was just about to say, that's why your work is so important. Do any of the flies with AFib also end up dying from strokes or heart attacks?
Yeah, so that's also a good question, and it's one of the reasons that we work on fruit fly. So if we were doing this in a mouse model, for example, and it had these kinds of fibrillatory cardiac events, the mouse would die, no question. But in the fly, they get their oxygen through a different system. They don't get it the same way we do through blood carrying oxygen through vessels. They get it through the equivalent of blood vessels, but it carries air directly from the outside to the tissues, and it diffuses directly across these tracheoles, we call them. And so because of that, the heart can be really bad, can be doing horrible things, but the fly lives, and we can study it. I mean, it's really hard to look at heart function in a fly who's dead, and its heart isn't working. So we can really study aberrant, badly working hearts in the fly because it doesn't die. And it will eventually, so there are some manipulations where the heart stops working completely in the fly, and they will die eventually because of that. But it won't be an instantaneous thing. It's mostly because the heart, its main function in the fly is to move nutrients from the abdomen to the brain and hormones from the brain to the abdomen for egg production and things like that. So it's basically a pump moving the blood of the fly around, but not through vessels. And it doesn't move the oxygen around the animal. So yeah, it doesn't have, I guess that fly, the mutant that I showed the video of, if you get them old enough, they have the fly equivalent of a heart attack where they just start fibrillating and they don't stop. They just basically shudder for as long as you want to look at them.
You did say that there's no real treatment for AFib. Somebody's wondering how effective antiarrhythmic medications such as dofetilide, I don't know if I'm pronouncing that right, how effective they are, and if those drugs work in your model systems, like the flies and cardiomyocytes.
Yeah, that's a great question. Yes, they do. We use them, and we've used them to verify that. So dofetilide, for example, is a drug that affects ion channel function. And it's one of the drugs we use to verify that in the fly we have similar ion channels as in humans. The problem with a lot of those anti-arrhythmia drugs is they have a side effect of arrhythmia, which seems crazy. But by affecting ion channel function, which is what they do, if you have too much or you don't have the right number of channels in your heart, you could tip the system, tip the balance from a slowed heart rate into a full-blown arrhythmic heart rate. And it's complicated to understand exactly why. I mean, even I know all about ion channels, and it's still very complicated to me to try to model how that works. But there are plenty of labs who do model the function of these channels and try to see how different drugs might affect the system. But in this case, the drugs are probably not going to be overly helpful. They are given, but they're not. You run the risk of having this side effect of increased arrhythmia if you're on the drugs. I think most of the treatments that are encouraged have more to do with modifying your diet and modifying and doing exercise within limitations and those sorts of approaches, at least in the early stages of AFib. If it becomes persistent, then meaning all the time, then that's when you have to have this electrocoagulation procedure done.
So if you catch it early and you make changes in your lifestyle, there is chances that it will not develop further and it might actually regress and go away?
You can certainly modify the progression. I don't know if it's possible to prevent it completely, but you can certainly modify the progression of as it develops from sporadic to persistent.
That's good to know. So the AFib genes that you are testing, they could also be drug targets, right? Eventually, hopefully, maybe.
Yes, exactly. And so, as I said, that one enzyme that we're studying right now, the desaturase enzyme, already has become a drug target for anti-obesity kinds of treatments. And so one of the things that we can do is pull it in and put it on some of our arrhythmia models, like that mutant I showed you, and see if we can reverse the arrhythmia in those models by feeding them the drug. So those are the kinds of manipulations we can do. Once we have what we think are targets, we can start going deeper into the research to try and find out exactly which drugs might work and might not. And certainly in the cardiomyocyte system, it's possible to you just dump the drugs on and can, in a very high throughput manner, test a whole lot of potential therapeutic candidates in a very short period of time. So again, it's a great marriage of these two systems that allow us to look at similar as well as somewhat different aspects of the problem. Because we can't look at aging in the cardiomyocytes, but we can in the fly, as well as diet. But it's a little trickier to screen through 300 drug candidates in the fly, but it's easier to do that in the cardiomyocyte system.
Yeah, that makes sense. And then you were talking about the genes, right, that could cause AFib. We've talked about diet and exercise. What are the overall risk factors? Are there any more that people should consider? And if your parents have AFib, you mentioned that when the parents have the wrong diet and have AFib, they give that to the next generation. Is that a for sure thing, or could it just maybe happen?
Yeah, so this brings in the concept of genetic background. So a lot of what a gene does depends upon all the other genes that are functioning around it, which is why looking at interactions is so important. And so a gene that causes a particular disease in one person might not in another person, because the gene that causes the disease requires other genes to exert its function. And those genes might be somewhat different in another person and may not interact in a way to cause the disease progression as it does in a patient. So understanding those kinds of, that's why understanding those kinds of interactions is really important to understanding the disease. And so no, if your parents have AFib, it does not mean you're going to get AFib. It means you may have a significant risk of getting it, and you probably should make sure that your diet and lifestyle is not going to exacerbate that risk. So you probably don't want to smoke, and you probably want to have a diet that's not got high in saturated fats and has lots of these mono and polyunsaturated fats, like in olive oil and that sort of thing. All of that is going to help with the progression or development of atrial fibrillation to begin with.
Thank you for that. And here's a question that you might have to help us understand. Does a flipped T wave affect AF? So I think that's atrial flutter. I'm not entirely sure.
So if a T wave is the part of the EKG that is the repolarization of the ventricle. Fibrillation, by definition, is in the atria. I don't know if there is any correlation to the repolarization in the ventricles. Primary defect is the loss of the P wave. I hope that answers your question.
I hope so too. By the way, I launched the poll. The survey poll, that will be wonderful. We have 2 more questions that we're going to go through, so there's a little time still to fill out that poll. One question is, what is fibrosis?
So fibrosis is a common response for many different diseases. So you can get fibrosis in your kidneys and and in spleen and liver. It's what it is essentially it's an injury response of different organs. So in the heart, for example, if you have a cardiac infarct where blood is occluded through the coronary vessels to a portion of your heart, that heart tissue will be damaged and die. And so what the heart does is then essentially try to make a Band-Aid to fill in that region where there's no contractile cardiomyocytes anymore. And it does it by making all these fibroblasts proliferate in this region. And they are not contractile, but they're essentially trying to fill in and repair this damaged portion. The problem is that often they don't stop, they kind of take over and you get a fibrotic response where you now have a bigger area of the heart that is non-contractile compared to the initial injury. And it's a real problem. And we would love to know how to make those fibroblasts turn back into cardiomyocytes in a human heart. We are not there yet. But it's mostly an injury response. But the bottom line is that it's non-functional in terms of what the heart has to do, and that is contract.
Thank you. And then the last question that we have is fibrillation controlled by magnesium and hormones such as menopause as a cause?
Well, there is an increase in AFib in women following menopause. I think in part, it's thought estrogen exerts a protective effect on heart function in women, which is probably why there's a lower overall incidence in women. And obviously after menopause, that's not there anymore. That's a good question. I have to think. So magnesium is involved in some ion channel function in the brain. I don't know about in the heart, but ATP, which is a major source of energy for muscle tissue, including the heart, requires magnesium in order to be functional. And so I don't know if there's a way you might have a magnesium deficiency that might lead to atrial fibrillation. I'm probably thinking about this too deeply, but it's most likely an effect of estrogen being absent after menopause. That is why women then start to develop more AFib.
Well, that gives me something to think about. Karen, thank you so much for joining us today. It was a fascinating talk, and thank you for all the answers and questions that you were able to go through with me from the audience. We've answered them all, which is fantastic. So.
Thank you so much for giving me this opportunity to present our work. We're very excited about it, and it's always nice to spread the word.
Yes, it is. And I find it highly fascinating. I hope lots of people will watch the video. I send a little message through the chat. This talk was recorded and we will put it on the Fleet Science Center YouTube channel on March 11th. I know that SVP, the Center for Bone-and-Crevice Medical Discovery Institute, will also put it out. So stay tuned. If you have friends who missed this, you are welcome to share it with them. Then also invite them to attend our upcoming talks. As I said, in April, first Wednesday in April, we talk about vision loss. And then the first Wednesday in May, we're going to talk about bones as we age. I hope you will join us again for this lecture series or any of the other events that the Fleet Science Center is putting out while we are still closed in this pandemic. But we're glad that we have the virtual world and the technology to still keep you engaged in science through video and Zoom and all the things that we have. And we appreciate our partners like SVP and Dr. Occor, who come join us and make sure that all of San Diego knows the amazing work that is going on in San Diego. With that, I thank you all and wish you a wonderful afternoon, and stay safe and stay healthy.
Bye, everyone.
Bye. Thank you.